The last organic molecules that we're going to talk about are nucleic acids. The nucleic acids consist of 5 component elements, which are carbon, hydrogen, oxygen, nitrogen, and phosphorus. There are 2 major classes of nucleic acids. The first is deoxyribonucleic acid, otherwise known as DNA. DNA is extremely important because it contains our genes, which affect all aspects of body structure and function. We will talk more about this in the next chapter. The building blocks of nucleic acids are called nucleotides. A nucleotide consists of a nitrogenous base, sugar, and phosphate. In nucleic acids, there are 4 different nitrogenous bases. These bases are adenine and guanine, which are classified as purines, cytosine, and thymine, which are classified as pyrimidines. The sugar group is deoxyribose. This is a 5-carbon sugar. It's called deoxyribose because one of the carbons is connected to a hydrogen and not hydroxyl group, which makes it different from the sugar ribose. Watson and Crick describe the structure of DNA as a double helix. Think of this like a twisted ladder, where the uprights of the ladder consist of alternating phosphate and sugar groups, whereas the rungs of the ladder represent the nitrogenous bases. In DNA, hydrogen bonding occurs between the nitrogenous bases, which is what holds the 2 strands together. In DNA, there is a specific pairing of nitrogenous bases. In this case, guanine and cytosine are 1 base pairing, and thymine and adenine are the 2nd base pairing. These are considered complementary base pairs. The shape of the DNA molecule maximizes the hydrogen bonding between these nitrogenous bases. Our other nucleic acid is called RNA. RNA is very similar to DNA in that it contains the 3 major components, such as nitrogenous bases, sugar, and phosphate. However, there are some key differences. First, the nitrogenous bases that are used to make RNA are different from those of DNA. While RNA and DNA both use adenine, guanine, and cytosine, RNA uses uracil instead of thymine. Additionally, the sugar used in RNA is ribose. This is why we call it ribonucleic acid. The other key difference between RNA and DNA is that RNA is a single-stranded molecule, which means it can form very complex shapes due to the interactions between the different nitrogenous bases within the molecule. There are 3 main types of RNA. These include mRNA, tRNA, and rRNA. There have been other types of RNA discovered and are categorized as regulatory RNAs. We will not talk about these regulatory RNA molecules in this course. mRNA, tRNA, and rRNA, along with those regulatory RNA molecules, are involved in expressing our genes. In the next chapter, we'll talk more about how this occurs within our cells. In your lecture guide, you have a small assignment. I want you to take the information we just discussed concerning DNA and RNA and fill out this table. Completing a table such as this, where you compare 2 closely related topics, is an active learning strategy that I encourage you to use as you learn about other similar topics throughout the course. The last topic for this chapter is ATP, which stands for adenosine triphosphate. This is the molecule cells use for energy. ATP starts with the nucleotide adenine plus ribose sugar. It then contains 3 phosphate groups that are held together with high energy bonds. An enzyme called adenosine triphosphatase is required to break these high energy bonds. When broken, they release energy, and this energy is used by the cell as fuel. ATP hydrolysis refers to breaking these high energy bonds and can be used to drive various reactions within the cell, such as muscle activity, cellular transport activities, and the formation of macromolecules through various chemical reactions. We will see the use of ATP as fuel for cellular activities as we go through the course. This ends our discussion of the chemical level of organization. I encourage you to create a study guide for this chapter using the learning objectives outlined in your lecture guide.